Estamos. Bien, bienvenidos a esta.
Welcome to our second panel session. I've been told to tell the students to be kind enough to sign the attendance list, then you will get a certificate of attendance. New aspects on healthcare services, from H1N1 flu virus up to the human genome. We're going to talk about the impact, the concept of a dynamic situation. We have here the know-how of outstanding Nobel Prizes laureates to talk about the current situation and the future. For many years, we've been reading about genoma. Sequencing human genoma was a real milestone, and at the same time, we learned many things about it. They were saying that by 2025, we'll start knowing something about it. That was said by Dr. Grisolia. And now you can get your own genome, and you can order this through the internet. New ways to look towards therapies, clinical aspects. Information technology has accelerated the study of the genome, and at the same time, globalization is like a snapshot communication. The patient is active, the patient is proactive, the patient has lots of viewpoints at the hand. It's like a resonance box with positive and negative aspects. As you all know, people, specialists, experts in the field of healthcare. Now it's public because of social networks, not politically speaking. We have also experts and different things, pioneers fighting against smoking habits, for instance. But prior to introducing the speakers, I would like to mention that Matt Redley said that, imagine, the genome is like a book, 23 chapters, chromosomes, each chapter, lots of stories, genes, it's gene with paragraphs, exons. Among exons, we have like ads, those are introns. Each word of the paragraph, it's a codon, and each letter, it's a base. And then the book has like a billion words, 800 Bibles. It is inside the core, the nucleus of a cell that has a lot of space. If you consider this the tip of a needle. So we have here 4 experts working in publishing this book of the genome. Here we have Mr. Arber, Werner Arber, who was born in Switzerland in 1929, microbiologist, Nobel Prize laureate in 1978 with Hamilton Smith at his side, working in the field of restriction enzymes. Restriction enzymes is the technology that has allowed slicing DNA to know where to do so and to have the recombinant DNA techniques, and studying also mutations, DNA mutations, and also inserts. Professor Werner Arber, where are we going in the field of genomics? Would you kindly describe where are we going in the field of genomics?
Well, within these few minutes in which I can talk here on this podium, I think I will not be able to cover everything. But I was at an early time in 1975 at the Asilomar conference. That was just a few years after genetic engineering had been successfully introduced as a research strategy, which helped us then to see new insights which were not accessible before in the genetic material. It also led a few years later to sequence analysis and so on and so forth. We will talk on this today. Now, at that moment in Asilomar, the conference was called, an international conference, to evaluate potential conjectural risks of doing so, of bringing DNA together which was not together before, recombinant DNA molecules. For example, taking a particular gene which you wanted to study, inserting that into a virus, then taking that virus, could be a bacterial virus, to infect a host cell and reproduce that gene millions of times in order to have enough material to really studying it carefully. That was very successful. Now, at that moment, the question of risks was really debated. And one had seen two types of risk of doing so. One, the gene, which of course you studied because you didn't know yet what was behind that gene, might have had some toxic or pathogenic effect on the researcher if you infect yourself in the laboratory by doing so. And it was concluded that this kind of risks should be, could be handled in the same way as the medical doctors when you are sick and they take a sample from you and then they analyze what is behind, bacteria, viruses. So, of course, we know how to handle that material. So short-term risk could be handled like that to study whether risks were really there. It was then argued also that at some later time, when recombinant DNA molecules were produced for applications and by purpose liberated into the free nature, for example, agricultural plants having modified genes, then, of course, one could have long-term risks which were evolutionary. Would these diffuse also out into other living beings than the ones in which they were introduced? At that moment, me personally, together with a few other colleagues, we felt that we should know better how normal evolution occurs. The other, by genetic engineering, would be human evolution caused by activities of human beings. At that moment, then, we started to see what can be how can we understand evolution, Darwinism at the molecular level? In other words, how do genetic variants come about in nature spontaneously? In textbooks, you see often, still today, that these are called faults, errors due to accidents. We came to the conclusion this is a completely wrong understanding of nature. In fact, we know today that there is a multitude of very specific mechanisms by which once in a while, in an individual of a large population, a genetic alteration is introduced. And normally, there are specific gene products involved, and there are also intrinsic properties of matter, like isomeric forms of nucleotides, for example, involved in the generation, spontaneous generation of genetic variants. And we came to classify these into so-called strategies of genetic variation. One strategy, we just heard that the human genome corresponds to an encyclopedia of about 800 volumes. That's tremendous. Bacteria normally are corresponding to one book. And one type of alterations in the genome are just one or a few adjacent letters, just one word is changed in some way or another. The other is that you tear off one page, for example, in a book, one of these books, and put it in somewhere, more or less randomly, at another place in the library. That's called transposition. And there are other rearrangements inside the genome which are well studied, and there are always enzymes behind. The third strategy is that you take one of these tiered off pages from one library, from one type of organism, and transfer it by horizontal gene transfer in another one. That happens in nature occasionally also, in bacterial world, in us, human beings, and so on. So this is acquired knowledge in the last very few decades. And this, in the session this morning, one talked about values. For me, I think this kind of insight into how nature actively makes biological evolution happening is of large cultural values for us. And it is also a good basis to evaluate evolutionary risks of genetic engineering. As a matter of fact, what the genetic engineer does is just applying one or sometimes two or even all three of these strategies. Local sequence changes within one word, rearranging, for example, an open reading frame, taking another expression control signal to have more yield of that product, or inserting a gene from another type of living organism. So therefore, I do believe that risks of genetic engineering are of the same order of magnitude as evolutionary risk in free nature and also in classical breeding of animals and plants.
Professor Hamilton Smith.
Professor Hamilton Smith shared the Nobel Prize with Professor Arber. He shared many of his theories. He's at present the director
The Celera Genomics Corporation, not only Nobel Prize, but Prince of Asturias Prize as well. And do you still think that with all this technology nowadays, we still are talking about medicine?
I'd like to take the opportunity actually to, is this working?
Yeah.
Okay. I'd like to take the opportunity to give the audience a brief history of the Genome Project and then where it's going in the future. In 1985, that's almost 2.5 decades ago, 30 scientists met in near Los Alamos in New Mexico to discuss the feasibility of sequencing the human genome and to decide how to approach the government to obtain $3 billion to sequence 3 billion bases of the human genome. I was present at the meeting, and there was a 2-day discussion, and what amazed me the most at the end of that meeting was that everyone unanimously agreed that we should go ahead with this project, even though the technological base for doing it was not there. Sequencing was being done by hand, and 10,000 bases was a big project for a laboratory. And the computational ability to actually utilize the sequence was also not developed at that point. Anyway, so it was decided to go ahead, and the first funding appeared in 1987, and then from DOE, Department of Energy, and then NIH came aboard a year or two later, and it was launched officially with money from the government in 1989. Things proceeded very slowly because, as I said, the technology was not there. It was not till 1998 that the first fully automatic sequencer was developed in one of the American companies. And when that technology was developed, that's when Celera was launched as a private effort to sequence the human genome, more or less in competition with the public program. And it was that competition that spurred the sequencing over the next 2 years of the genome. The draft sequence was completed just 2 years later from the appearance of those fully automated sequencers. So it was the technology that finally caught up with the dream of the scientists and propelled it forwards, and also computational power continued to develop in those years so that they all came together to give us the human genome. So 2000, now 9 years later, the sequencing technology has continued to advance so that we have the sequences of thousands of different organisms and several different human genome sequences. And I think scientists now predict that the technology will continue to develop so that within another 5 years, it will be possible to begin sequencing every individual person. In 10 years, perhaps, it may be possible to sequence each child at birth to obtain their complete sequence, and that will launch genomic medicine. Of course, we'll have to correlate the phenotypes of many individuals with their sequences to really be able to read what each person's genomic sequence means in terms of their susceptibility to diseases, cancer, how they respond to different medications, and so on and so forth, which will be the basis for our genomic medicine.
Besides, Professor Hutchinson will be his investigator at the Craig Venter Institute, and he's much more interested in improving methods to learn about gene function in DNA. And he's one of those convinced that before 5 years, science will be able to make a virus, something alive, out of nothing. And not only that, about the future of something I would like to have described, the minimum genome.
Okay, thank you. And thank you, Professor Calvo, for this wonderful event. Virus genomes, a number of virus genomes have been synthesized from chemicals. Some simple things like bacteriophage genomes, a DNA that codes for poliovirus genome has been synthesized. The 1918 flu virus genome has been synthesized. And this isn't making a virus from all non-living parts, because to get a virus from these synthetic genomes, you need to introduce the DNA into a cell where it can then initiate transcription, translation of the virus gene products and assembly of virus. But these things have been accomplished, and in a group led by Ham that I'm part of at the Venter Institute, we've synthesized genome of a very simple bacterium, the mycoplasma. Mycoplasmas are bacteria that are simple, not because they're primitive, but because they are minimal. They have lost a lot of unnecessary things, like in the movie last night. They have lost a lot of things that they don't need in the situation they find themselves. And so we're interested in studying these minimal genomes that have just a few hundred genes. The smallest naturally occurring genome of a cell that can live by itself is about 500 genes. And we're trying to see if we can reduce that substantially. And our main motivation is not practical, but is to try to understand the molecular basis of life. So we would like to have a system that's simple enough that we could model it computationally and be able to predict from a genome sequence what the products are, what proteins they make, what those proteins do, and so forth. But we're a long way from this. Even with the simplest genome, about a third of the proteins, we have a very bad idea, if any idea, what they do. And the situation with humans, of course, is much worse. So we have a lot to learn.
Talking about things that weren't understood at first, works of Professor Richard Roberts have a lot to do, his Nobel Prize has to do with that, with the discovery of the introns. Is that right? At first they were considered something useless within DNA. Nowadays, some people consider them part of the future as well. What is your position on that?
Well, even though I discovered them, I've not worked on them for an awful long time. So I really take the position of an outsider looking in, looking to see what everybody else is doing in this field. And I think it's quite clear that whereas we originally started off thinking that they were junk or trash or something that was unnecessary and not useful, we've already found that there is quite a lot of useful information inside this so-called junk DNA. We know that there is a lot of regulation that takes place there. And perhaps one of the most exciting findings recently concerns not the introns per se, but the fact that there's an incredible amount of transcription that is taking place in the human genome and in other genomes that is certainly regulatory in nature, but we don't quite know how it's doing the regulation. And so I think one of the things that comes out of all of this is that introns really told us that RNA was very important inside cells. I think most biologists believe that the very earliest genomes, the very earliest genetic material was all RNA, and that at one time RNA was also doing a great deal of catalysis and is still doing catalysis. For instance, we now know that the ribosomes that make protein, it's actually the RNA that is catalyzing the reactions. And so I think there's every reason to think that as we get further and further into trying to understand what's going on in the human and other genomes, we will find that RNA is playing an ever-increasing role. Maybe even it, too, is catalyzing things inside the nucleus, perhaps inside the cytoplasm, that we just don't know about at the moment. So I think if I were a young biologist starting over again, I would think that RNA was a very attractive subject area, something where you know there are going to be big discoveries and where maybe there's another Nobel Prize or two lurking.
Thank you. Si estábamos viendo...
Now we see life through the microscope, and now we're going to analyze the macro situation. Horacio Rubio Monteverde, Director of the National Institute of Respiratory Diseases in Mexico, full professor at the Autonomous University from Mexico, an expert in the field of smoking habits. He has followed in Mexico the latest de novo discovery, or the first one this century. Can we talk about the Mexican flu, or we should stop using this term? What do you think about it?
Fue de los primeros y hubo
I think he was amongst the first. Well, I think that it was premature knowledge, which was the basis for some measures, which now we see are extremes. For example, the emergency, the alarm, which was triggered in April, schools were closed, etc. And sometimes some people would call us Mexican flu, and now there is the influence of H1N1. It has had a big impact on the world because until November 15, the United Nations said that there were 550,000 cases and 6,700 people died. And in Mexico, there were 60,000 cases and 573 people died. And now, with a wider knowledge, we concluded that it had been an extreme measure, but we didn't know that then. And this is why the Minister of Health took that measure, and we thought it was important. But if we take a look at the diagram now, there are more cases today, but mortality, although we've seen that more or less 200% of the cases are hospitalized, and some of them die in UCIs. with a very quick and dramatic death, because in most cases they are combined with pneumonia in intensive care units, and they present dramatic situations with hemoptysis, in many cases, like in all cases of TB, of tuberculosis. I think, therefore, that these aspects were quite or drew people's interest, but now there has been a decrease. Some epidemiologists, however, believe that late this month and in January there will be a third breakthrough or outbreak. We hope it won't be so strong, and we have taken some measures. Albert Jovell, who's there, is the creator of the Spanish Patients Forum. Albert Jovell has said, patients want to be cured, healed. I accept that I may not be cured, but it is difficult for me to accept that I'm not taken care of, because, well, he is not only, and he's not just a patient, he's also a physician. Is the medicine of 21st century dehumanized?
I think we have progressed along the last few years regarding technical aspects, and considering, for instance, that nowadays what has been presented here, that information confirms what I'm saying. Access to science and technology makes us not focusing on what is the most important thing, a relationship with our patients. We'll have to get it back, and that is part of a training and education we'll have to give to our professionals, empathy and good relationships with patients. Now, young doctors, for instance, sometimes they forget about clinical history, the physical exploration, examination, just what we said before, palpation, or using literally a dialogue as a tool to learn from patients, because now it's more complex. And I think that what has been presented here, the Mexican situation in the case of the flu virus H1N1, it is what we're witnessing here, not only in the case of the flu, but also in multiple pathologies. We have a generation of patients having several conditions at the same time: diabetes, cardiovascular risk factors. They might have had cancer and got cured. Those are patients getting multiple drugs. If we give one drug, there are side effects, and a combination of drugs is different. So we'll have to focus on human relations as something requiring time and exploration. Javier Toledo, as a general director from Aragón, he is in charge of campaigning against smoking habits, also against AIDS. And there are things that are not related, stricto sensu, to health. Europe nowadays, in Europe we have less smokers, but in China people are smoking more. Global health, what does it mean if you reduce 1% of Europe, if you have 1% of smokers in China? The situation is getting worse. What do you think about it?
Allow me to thank Professor Calvo and his team and the sponsors for allowing the organizers to hold this Congress. A Congress of these characteristics, of course, must be funded by a sector in the economy of market, and this is thought-provoking. Going back to the subject that worries me, smoking, it can be considered as the hidden plague. Flu has caused 1,000, 2,600 cases, for example, in Spain. But in Spain, every day 150 people die from smoking, which means that there are 6 people per hour dying, 1 person every 6 minutes. And we're talking about not a disease, about an addiction, because those pathologies can be avoidable. But compared with other pathologies, for example, the flu virus has to find his way ahead. Well, smoking has economic connotations that we should analyze. We're not fighting against a virus, but against an epidemic which has been caused by man. and of course maintained by many economic interests. Anyway, the developing country, in the developed countries, for example, Spain, the epidemics is decreasing, but unfortunately this epidemics is moving to developing countries, and this entails more health inequalities. We're talking about countries where having access to drugs is very difficult. Tobacco and cigarettes are sold very easily. Advertising is permanent. We should use many strategies other than prevention and therapies. Begoña has always has been in charge of public health in the autonomous community of Galicia. And the media sometimes work in cooperation with institutions in campaigns. The independent social media have an increasing role to play, for example, the internet. To what extent do social networks have a role to play, and they're playing it? Well, I think that the social networks are now one of the key points that allow us to work in subjects related to risk factors for chronic diseases, everything related to lifestyles. We have mentioned smoking, the need for devoting more time to patients, and working with them. We should also be talking about the problems which are being generated today by overweight and obesity. This morning, somebody said that hunger is killing lots of people. But at the other end, in the developed countries, the way we eat, the eating habits are killing thousands of people. So working with patients' organizations and all kinds of citizens' organizations is very important. For example, in Galicia, when we started to work in the area of smoking in 1993, we started working with society through a network of promoting centers. At that time, there were no restrictive laws, and numerous centers, including citizens' organizations, professionals, etc., got involved in control or in fighting against smoking, and that was a very important platform to disseminate our activities and to have more participation. You asked before if a prize, for example, inviting people to spend holidays in the Caribbean would be an incentive for people to quit smoking. I think that it would, together with other strategies, that would be an incentive so that people establish a day to quit smoking, commit themselves to fight against smoking for some time. And, well, we carry out actions through biannual activities, and we have achieved decreases of 30 to 35%, which is a very high figure when talking about smoking. And working with those networks allowed us to disseminating our actions much better and to have more participation in our strategies. You know that the basis of this campus is the involvement of attendants, and because audiences are requested to ask questions to the speakers, to the panel, or to other experts. This is why we're going to give the floor to the audience. If you want to ask something to Dr. Jovell, please start with him, because he has to go very soon to a lecture. But I would like to ask Professor Arber if he thinks it is foreseeable to see acceptance of genetically modified organisms as a way to modify therapeutic treatments, for example, or as a way of having those treatments.
Well, yes. Some minutes ago, I told you that we came to conclude, yeah, it works, that conjectural risks of using genetic engineering are of similar order of magnitude as happens in nature all the time. And we do know that in biological evolution, we also know that in classical breeding techniques, very few, if any, disaster has happened in historical times, which we can have noted. Therefore, at least as long as no artificial human-invented genes are used, I think genes which are already in nature have a certain chance occasionally to be transferred to other living beings. We know this very well from important studies with microorganisms, bacteria, and their viruses. As a matter of fact, we do know how, by which enzymes and which gene products, genes, for example, for antibiotic resistance, are transferred horizontally to other microorganisms which are nearby. We also know that some bacteria have what is called pathogenicity islands. If they transfer to a non-pathogenic bacterium, they may render that other bacterium more pathogenic. So all of that is happening in nature. But don't believe that only these islands, short segments of genetic information, which are of medical importance, are transferred. Any type of chromosomal gene can be picked up by a gene vector or by other means and being transferred. If I say occasionally, that's rare, very rare event. And of course, in evolution, none of the novel genetic variants, none of the novel mutants are targeted to fill identified needs. In most of these cases, we know that there is at least some randomness of what's coming out. And that, of course, does make sense for biological evolution, because living beings do not a priori know when and when they will come, in which environment, in which they need different capacities. And those few living beings which have suffered a mutation and are mutations which are favorable for novel living conditions are, of course, the winners. But most of the mutants will be either rapidly or very slowly again be eliminated from populations. So with that, I want to come to your question. I'm fully believing, and I actually propagate that idea, that with our available now and within the next 2, 3, or 4 decades available information on functions of genetic information, we will probably be able to modify a dozen or 2 dozens of our normal agricultural crops, which we provide our daily food, in such a way that these plants are more rich in those daily ingredients which we need to take by nutrition. Vitamins, minerals, essential amino acids, and so on and so forth. With metabolomics, genomics, proteomics, we get daily input of what we really know better and better, what is the need. And in knowing that, I think it will be possible within the next decades, not tomorrow, but within a few decades, if the will is here, to construct plants which are healthy as normal food for our daily diets. These plants can also be made more healthy themselves, that they don't get rotten during their growth already or during their storage. And some of the plants can use soils which are perhaps rather dry, some others in wetlands and so on. That is in the feasibility.
De alguna forma...
and in doing so, I think we will have, this morning we had, that's just my last conclusion, we heard about malnutrition, we heard about hunger. I think if you follow these possibilities, you will have an extremely good way to fight against these problems. And we should be able, with our limited water supply, with our limited fertile soil, and with respect to maintaining big biodiversity, to have enough food for every person on that planet.
Jovell, un paciente reconocería
A patient will then be able to see that clinically as a treatment. Well, that's complex because patients, first of all, a patient is a very vulnerable subject. A patient is expressing signs and symptoms, and the patient wants practical solutions. When the patient is told about what might happen, there are contradictory feelings. On the one hand, expectations and hopes, but then the other situation that the patient knows he or she is not going to benefit from all the discoveries. On the other hand, some discoveries are like what we wish to have, like wishful and thinking, but not always something that in daily practice will be translated into clinical applications. Sometimes what we want from patients, and when we talk about research, we should be extremely cautious to take it with a pinch of salt. Since the 90s, we've been told that there are going to be vaccines against AIDS, or, there was recently something in Thailand, but it's taken 25 years, and we'll have to be extremely cautious when we talk about expectations given to our patients. A question to Professor Smith.
How will we interpret the vast information in someone's genome and the complexity of that information? For example, in the epigenetic regulation and things like that. How will we interpret the vast information in someone's genome and the complexity of that information in this era of personalized medicine?
That's a very good question. What we have to do is to have the genome sequence of a large number of individuals and correlate that with each of their phenotypes. In other words, to make sense out of the genome, to know which version of a gene is correlated with which effect or phenotype of that individual is what we have to learn. Once we have obtained all that information, which is dependent, of course, on having a lot of human genome sequences and a lot of medical data on those individuals, put it all into computers and make the correlations, and then we can take somebody else who has a sequence, look at that sequence, and on the basis of all that information we've gathered, we can now make predictions of for particular genes. If it's a version of a gene that has certain base changes, that would mean that that person perhaps is more susceptible to a side effect of a medical treatment, or that treatment might be very good for that person, and so on. So it's a matter, it's going to take decades probably to get the proper information.
Another question for the whole panel is about the controversy on genetically modified crops. On one hand, it's clear that you believe in that, and it's clear there's a frontier in between both sides of this, which is called the internet, somehow. We all know in this country the story of a nun who is able to take away all the information of the influencer, and no matter how much you say how good they could be, there's always a sensation that they could be bad. What is your opinion about GM crops and how they may change the future, both in the medical, from the medical point of view and from the economical point of view and agricultural point of view?
I think genomic medicine of the type that I've described will have a huge economic impact, because we'll know right away from a person's genotype what medications are best for him instead of experimenting. Now we have several different drugs for each treatment, and you give a patient a particular drug and see how he responds to that, and then maybe you have to change the drug because he has a bad side effect, and so on. So it would point to proper treatments, and also an individual's response to different diseases could be predicted, perhaps.
But not only medicine and diseases.
At this point, we can't really foresee all of the implications.
In crops, they are going to bring us new, improved agriculture and functional products more and more every day.
He's asking about crops. Yes, I think, well, of course, there are many genetically engineered crops, and they're in use, I guess, more in the United States than in some places. And there's the potential to, there are rice strains that provide a much better balance of amino acids than the
golden rice,
natural rice, and that can save many lives due to malnutrition. I think, as was pointed out in the first session, there's a lot more than the technology that's involved in getting these things out and in use. And many of the world's problems are not limited by the technology, but by the ability to use that technology politically and to distribute things.
¿Y los recursos cómo afectan?
Resources. How about resources in Mexico, for instance? South America is a good example. To what extent are priorities giving in fighting, for instance, smoking habits or dengue? I think there is an important factor there, and we'll have to get into the core of the subject. But that is very difficult because countries, in order to reach an agreement, for instance, the United States, they spend 19% of the GDP in healthcare services, and 50 million people are not covered by health insurances. Other countries like ours, we have less than 6%. 50% of the population have right to social or healthcare insurance, and 50% are deprived from healthcare services. Same thing in other countries. With what United States are investing, we could cover all Latin American citizens, giving universal healthcare coverage that was described by Dr. Marno. But I think that costs or what we spend in smoking in Mexico is a limited amount compared to 160 persons dying per day, like here in Spain, and that's a big problem worldwide, and this is motivated by a terrible situation, and it's like the evil aspect of it. And I think that we should enhance our knowledge, our global knowledge, and we should say, well, why is it so that some people are so well off and some other people are getting less, but 8% of GDP is enough to give coverage, and I think that we should enhance that, and we should focus on that at a fora like this, organized in a very important way and we've been here for 5 years, and we thank Dr. José Ramón Calvo. I think that we're going forward, but we need to enhance knowledge in the field. Sometimes people are allocating resources, but we are also considering social networks, to what extent prevention campaigns, public campaigns, are getting the public segment or the target group. Sometimes it is better to have an NGO group working in the field. That happened, for instance, they disseminate the message when the administration, when they try to get into Facebook, they don't know. You have to put it into a social fabric. Well, we have to get alliances, and messages should be the same from different supports. That's going to multiply responses to the action. Of course, I think that sometimes, yes, you need to get external contributions in order to disseminate the message and to deliver the message to the population, but we should also assess the type of messages we're conveying. I think that sometimes we take a distance from the population. We work at the office and we've lost gears. We've lost a total approach on the population we're dealing with. We think they are like we think they are. They understand what we tell them, and they live like we live, and they have our levels. But sometimes we adapt the message, but not always, and we do not assess the impact on the population. I would like to say that we'll have to measure if the campaigns are or not understood by the target population. So we'll have to see how to draft messages to see if we can convey messages properly and if they are understood properly. We have to get alliances within the social fabric to disseminate the message in the proper way and to have the proper penetration. I don't know how members of the panel, they do see the fact that a condition that is simple like the flu is meaning a threat worldwide. What do you say about it? A case of H1N1 flu virus that is like, for instance, other viruses, other types of flu. Would you elaborate on that? I think that here we have an aspect that is very important, and our Nobel Prize laureates might elaborate on this. If you remember, at the time of Gerald Ford, there was a situation of epidemics of a sort. Population, well, people were vaccinated, but when they arrived to a certain figure, they saw that side effects were so that they stopped vaccination campaigns. And one of the aspects, they said, on top of technological knowledge and progress and what has been mentioned by outstanding professors here, it is something that's been mentioned. It is a fear. People do fear. And you see that some people do not want to get a vaccine. And that's the same situation in Mexico, with the availability of the vaccines. I think we should give information, and that is related to the previous roundtable. Education is fundamental, and medical education plays an important role, and it is how to achieve what the lady doctor said, how to convey this information and disseminate the information. But that is, there is not a grey cap research and society. Would you, a grey or zone research and society? Would you elaborate on this, Professor?
Well, let me just say something about this flu virus. You may note that these are absolutely interesting examples of biological evolution. We know, we are told, and we believe so, and we know that it is so, that they mutate all the time. It's enough that a few individuals become mutants, to become more aggressive, more pathogenic, and those find their hosts and will multiply then, and then all of a sudden the epidemic comes up. That's why we have to be vaccinated over and over again every year against the expected type of virus which are around. They are not stable. And I think this contributes if we instruct the general public on also the way of evolving biological functions. I think that will help them to understand what's all about. And we encounter now, for example, possibilities within Europe, in my country, that quite important fractions of the population refuse to become vaccinated, just in fear that some side effects may happen. I think this is also a problem. Of course, we cannot claim that there are no side effects. It depends on the particular person. So we are not yet having sufficient knowledge who could best be vaccinated against that particular virus and so on and so forth. But we are in the process of learning. I think this is important, that we see here the connections between these two things.
Yeah, vaccines are fascinating, and not everyone has to be vaccinated to wipe out a disease. An example are the pneumococcal vaccines that are given to small children. And this is a vaccine that is directed against several specific antigenic types of pneumococcus. And what's been found is that although it was given almost entirely to small children, those types of pneumococcus are not found anymore, even in people my age. They're extremely rare. This, of course, is because children are a real vector for disease in human populations. So often you can get rid of a disease by vaccinating part of the population. About H1N1, I think I had it. I think the number of cases is very much underreported. I have a 5-year-old son who had the flu, and when he had the flu, I had a sore throat. I felt a little under the weather for a day or two, and that was it. And I'm convinced I had H1N1. My wife had a much worse, she's younger, she had a much worse sore throat, but she was okay. But these are cases that are, of course, not reported at all. I bet it's underreported by a factor of 5 or 10.
Begoña.
No, quería volver a enlazar un poco con...
Same line of thoughts, what you've said at the beginning of the presentation. Why is it so that people refuse to get a vaccine? I think that there was uncertainty when the first cases started, and there is sort of a direct dissemination of the information through the media, and people are having doubts, the same doubts we people have, the administration and healthcare professionals, when they talk about the possibility of getting a vaccine or not, if we're going to have time or not, if we're going to get all the tests, if this is safe or not, if we're going to have a vaccination of the population. Now we say, no, pregnant women shouldn't. And now we say we have sort of an adjuvant solution. And people take it with a pinch of salt. They have reluctancy. And as we consider that the effects are not as bad with a very high mortality as was foreseen, and we see those are very mild cases, it's understandable that people are not committed to getting the vaccine. Javier, I'd like to take into consideration the H1N1 flu virus in the light of the objectives of our campus of excellence, university, companies, and the goals for the millennium. I think that we've been effectively managing a disease that was not existing. I think that healthcare authorities and World Organization, they've worked together in order to solve the problem of H1N1 flu virus. It was not as important as we thought at the beginning. I'd like nowadays to take the objectives for the millennium. Out of the 8 objectives from 2000 till 2015, there are specific goals regarding healthcare services, fighting against poverty, improving health and development. But we have child care, maternal care, and diseases like malaria, like HIV, AIDS. So I insist, perhaps the example of a global response is to be applied to something we have here in the objectives that were stated worldwide, objectives for the millennium, and we are putting them aside perhaps because we are focused on problems that are not as important. I would like to go back and insist that from our gatherings we should try to say it again, that the World Association for Development should take place in order to improve healthcare services worldwide, having access to drugs that countries cannot buy. And trade is very important, fostering, for instance, poor countries' economies, and access to new technologies, which is fundamental. We should insist on macro projects, but that makes me think that the flu is a good example that we have to focus on important things we are sometimes putting aside.
I would like to say a few things that some may view a little controversially. One is, I think we've learned something rather important from this latest flu situation. One is that the media is pretty much totally out of control. They blew it up, they thought it was a great entertainment story, and they produced a lot of responses that were not the best. Okay, so that's one thing. The second thing is the politicians also thought that this was something rather good to get on the bandwagon with, and I think we've had a lot of misinformation that has been put out by the politicians. Third thing is that we also should have learned, and I fear that we probably haven't, that growing the flu virus in eggs is not the best way to make vaccines. We know of many better ways to make vaccines, and frankly, growing these viruses in eggs is not the way to do it. You can never produce enough vaccine in time, and I think that's been shown perfectly clearly. So I would like to think there should really be a wake-up call that we should go back and take a look at what's going on and figure out better ways to do it. And there's something else that comes out of all of this too, and that is that commercial companies in general are not very interested in vaccines. They'd much rather make Viagra. Okay? Vaccines have the disadvantage, as far as they're concerned, is that they actually have an effect. They work. You don't need to keep taking vaccines over and over again every day, whereas Viagra, you do. And so I think we should also think as a society, what is it that we want? The very best medicine that we have going after aspirin are vaccines. Vaccines work. Sure, there are a few side effects, but we don't live in a risk-free world. Politicians, the media, would very much like you to think that you live in a world where you can remove all risk. The answer is you can't. And in fact, if we did live in a world where there was no risk, it would be a very uninteresting, uninviting place to go. I don't want to live in a world with no risk. A little risk is a good thing. Anybody who's been down a ski slope knows that a little risk is a very good thing. And I think we really should look upon this whole business as a time to reevaluate what is important to us as a society. What is it that we really want to get out of medicine? Are we really doing it the best way? Is private companies dealing with these problems? Is this the way we want to go? Is this how we should be making vaccines, where the profit motive is driving everything? And finally, should we let celebrities from Hollywood be telling us that vaccines are dangerous and scaring half the population?
Professor Smith.
Yeah, just a brief comment. I totally agree with Dr. Roberts about the importance of really promoting vaccines. The immune system is the only other type of cell in the body other than the brain cells that can learn things. And vaccines essentially put the immune system to school and train it. But I think, I don't know for a fact, but I think that the FDA may play a role in the fact that they still have to make them in eggs, because that's an approved procedure.
Exactly.
And we have to get on the ball and start doing it by better methods. For example, producing the actual proteins in coli or some other organism.
There are other ways in which the drug companies influence what medicines we have. For example, we don't have any really new classes of antibiotics in quite a while. And as I understand it, the major pharmaceutical companies in the U.S. have essentially dropped their ongoing efforts to produce new antibiotics. And
but you know why?
Because the important diseases are diseases of poor people.
No, it's because they cure diseases. And that's not what the pharmaceutical companies want. Okay? As soon as you've cured a disease, you've lost your market. And so this is why they like to provide drugs for chronic diseases, where you have to take the drug for the rest of your life.
Okay. I think there's also a part of it is that there isn't a lot of money in better antimalarials, for example, or such things.
But shouldn't genetics be looking into new vaccines as well?
Yeah, yes.
Synthetic vaccines as well?
Yes, yes. And there's work in that area. And there's, you know, work in academic labs related to antibiotics too. But you really kind of need big, it's very expensive to get an antibiotic out and get it tested and in use. And that's not something that can really happen effectively in academia.
Do you agree in the end that research and education are the two main vectors to wipe out any disease?
Yes, I think so. I think it would be nice if well, as scientists, we aren't always
You don't only need the vaccine, you need the general public to acknowledge it.
Yeah, we need the general public to understand what's going on. That's in large part maybe our fault for not communicating it well, and the press's fault for not communicating it well in all cases. I think news coverage of science is better than it was 20 years ago, maybe, but it's not
enough.
Not wonderful.
Maybe what we need to do is to get the help of the advertising industry, because they seem to be able to sell anything. They were able to sell cigarettes for an awful long time.
Professor Arber.
Well, I think my proposal, which I'm going to make now, I know it's difficult, but it can work. With all these questions, drugs for use ones which work and so on, there are other things, GM crops and so on, where much of misunderstanding from the scientific point of view, scientific understanding from the general public is inhibiting applications, then money-oriented producers of some aspects. I think what could help in these situations are public-private partnerships. That means politicians, as speakers of the general public, could take an initiative to group together with companies which have know-how and fundamental scientists who produce know-how. And I think if we could do that, that could help case by case, GM crops on one hand, these drugs on the other hand, which were here criticized and so on. I think that could be, in many cases, a big help. It will not work from one year to the next, but it will work within next few decades.
Bien, ¿alguna pregunta más?
Thank you. Questions, if any, now with the microphone. I think it is about time to make conclusions and to say that science, of course, has a word to say, but education and society should go hand in hand, understanding science to make knowledge help people to live longer and to live a better life and a happier life. Thank you very much for your kind attention.